Vanakkam friends, the topic of discussion for is on germplasms and its conservation, evaluation, documentation, distribution and quarantine. I welcome on behalf of Trinity Cultural Academy Coimbatore and New India Publishing Agency New Delhi for this presentation. And this is being prepared by Dr. Vanangamudi, formerly Dean Agriculture of Agriculture College and Research Institute, Tamil Nadu Agriculture University, Coimbatore. And coming to the topic in particular, this is the germplasm conservation. What do you mean by that? It is the protection of genetic diversity of crop plants from genetic erosion. Erosion, that is more important. There are two methods of conservation. One is in situ conservation and the second one is ex situ conservation. First, we will discuss on in situ conservation. It is conservation of germplasm under natural condition. This is more important and this is being achieved by protecting the area from human interference, not only human interference and also the animal interferences. And the examples for in situ conservation is natural park, biosphere reserve and gene sanctuary. And the National Bureau of Plant Genetic Resources in New Delhi established gene sanctuaries in Meghalaya for citrus as well as in northeastern regions for Musa, citrus, Oryza and Saccharum. And let us see the benefits or the advantages of this in situ conservation. It is a wild species and natural, semi-natural ecosystem are preserved together. This is more important. As far as the demerits are concerned, there is a larger area or several area has to be allotted for conserving a single species. And the management is very difficult from natural disasters and also human interference and animal interference, and it is very expensive method. Next conservation technique is the ex situ conservation. It refers to the preservation of germplasm in gene banks or seed banks. The advantages are it helps to preserve entire genetic diversity of cross species at one place and very small area is needed when compared to in situ conservation. It is very easy to handling and it is the cheapest method. And as far as germplasm conservation is concerned, there are 5 ways of conservation. And let us see one by one. The first one is seed banks. It is the germplasm or seeds are stored of various genotypes in one place. If it is orthodox seeds, it can be dried to low moisture content and stored at low temperature. The examples for orthodox seeds are maize, wheat, rice, carrot, papaya, pepper, chickpea, cotton and sunflower. Most of the leguminous crop seeds also. And it is possible to store only orthodox seeds in seed banks. But it is not possible to store the recalcitrant seeds which is having high moisture content for maintaining the viability about 35 to 40%. If you decrease the moisture content to 12 to 13%, there is a drastic loss in viability. So it cannot be stored in the seed bank. So the best examples are citrus, cocoa, coffee, rubber, oil palm, mango, jackfruit and as well as date palm. And there are 3 types of gene bank conservations. One is short-term conservation. It is nothing but the samples are stored for working purposes. It is stored very short time especially for 3 to 5 years at 5 to 10 degrees Celsius. And regarding the medium term storage, it is active collections that are stored for medium term of 10 to 15 years at 0 degrees Celsius. With respect to long term, it is the base collection that are conserved only more than 50 years or more at minus 18 degree to minus 20 degrees Celsius. There are many advantages as far as gene banks are concerned. Large number of samples can be conserved in very small space, handling easy, free from pathogen and insects. We can protect all the seeds from diseases and pest attack. And regarding the disadvantages, as I told earlier, recalcitrant species cannot be stored. And if there is any frequent power supply failure, that may lead to loss of viability, but it needs periodical evaluation to test the viability, seed viability, especially the germination percentage. After some time, you have to multiply all these seeds, and then fresh seeds are stored again to maintain the storage period, to extend the storage period. And the second way is plant bank, is nothing but the field bank and areas of land which germplasm collections are growing plants are assembled in a natural conditions. There are some limitations. It requires larger area. It is expensive to establish, not only establish, but also costlier for maintenance. It prone to damages like diseases and insect attacks. It is a man-made one, and it also prone to natural disasters. And there are human errors in handling the samples. That is more important. And we have in India, we have established field banks, and not only in the India, throughout the globe. And I can quote few examples. In the country Malaysia, about 500 hectares of oil palm is being conserved. Indonesia, 1,000 hectares areas for coconut and other perennial crops are allotted for maintaining the genotypes. And Philippines, that is Southeast Asian germplasm for banana is being conserved. In India, global collection of coconut in Andaman Nicobar Island, and they are and in that place these coconut collections are being conserved. And the third way is shoot tips, nothing but the meristems, meristem cultures, and not only meristem but also nodal segments are also being conserved. And advantages are, it if you conserve the shoot tips or meristem culture, we can get virus free and pathogen free materials. And mostly this shoot tip or meristem culture is especially used for vegetatively propagated crops like potato, sweet potato, cassava, that is tapioca, and regeneration of meristem is extremely easy when compared to previous methods, and recalcitrant seeds can be easily conserved by meristem cultures. And the fourth way is cell and organ culture. It is nothing but the cryopreservation, storing the embryogenic cell cultures and somatic and zygotic embryos in liquid nitrogen at minus 196 degrees Celsius. That is liquid nitrogen. And the fifth last way of conservation is DNA banks. The DNA segments from genomes of germplasm accessions are maintained and conserved. And next we will see the gene banks that are available in India. And for all crops, the National Bureau of Plant Genetic Resources located at New Delhi is conserving the all crops, and it is established in 1986. The Directorate of Wheat Research have germplasm collection of wheat. It is located at Karnal and established in 1965. Rice germplasm is conserved in Central Rice Institute, Cuttack, established in 1946, and potato germplasms are conserved in for Central Potato Research Institute located at Shimla. It is established in 1945. Regarding pulse germplasm, it is being conserved at Indian Institute of Pulse Research at Kanpur and established in 1966. And Directorate of Oil Seeds located at Hyderabad and established in 1977, and it conserves all oil seed crops. And National Research Center for Sorghum that was established in 2009 at Hyderabad. They are maintaining the sorghum germplasm. Now this National Research Center for Research was upgraded as Indian Institute of Millet Research in 2014. And regarding soybean germplasms, National Research Center for Soybean at Indore is conserving and established in 1987, and Central Institute for Cotton Research at Nagpur, they are conserving the cotton, and it was established in 1976. Central Plantation Crop Research Institute at Kasargod, established in 1916. They are maintaining plantation crops. The tobacco germplasms are maintained at Central Tobacco Research Institute, Rajamundry, that was established in 1947. Tuber crops other than potato that are being conserved in Central Tuber Crops Research Institute located at Trivandrum, established on 1963. The Indian Agriculture Research Institute located at New Delhi, established in 1929. They have germplasm of maize. And regarding the forage and fodder crops, the Indian Grassland and Fodder Research Institute at Jhansi, established at 1962, maintaining these crops. And with respect to groundnut, pearl millet, sorghum, pigeon bee, and Bengal gram, it is being conserved. They are being conserved in International Crops Research Institute for Semi-Arid Tropics at Patanjeri, Hyderabad, established in 1972. Even though it is an international institute, since it is located at in India, so I have included this in Indian institutes. And the Forest Research Institute that was established in 1906 at Dehradun, they are maintaining all germplasm of forest species. And Botanical Survey of India, very old institution that was established in 1890 at Calcutta, they are maintaining the germplasms of all crops. That's all about the germplasm institutes at India. Now we can see some of the international institutes that are conserving germplasm. And IRI, International Rice Research Institute, Las Bonas, Philippines, established in 1960. They are maintaining the tropical rice. There are about 42,000 rice collections are there in IRI. And CIMIT, established in Mexico during 1966. They are maintaining maize, wheat, not only that, triticale, barley and sorghum. There are about 8,000 maize collections are being conserved at CIMIT. CIAT, established in 1967, and they maintain the cassava, beans, also maize and rice in collaboration with CIMIT as well as IRI. IITA, International Institute of Tropical Agriculture, Ibadan, Nigeria, and established in 1967. They maintain the grain, legumes, roots, and tubers as well as farming system. The CIP, the Potato Research Institute, established in 1971. They have germplasm collection of all potatoes species. And the West African Rice Development Association, Liberia, and established in 1970. They have a regional cooperative rice research in collaboration with IITA and IRI. IBGRI, International Plant Genetic Research Institute, located at Rome, Italy, and established in 1974. They have genetic conservation. AVRDC, the Asian Vegetable Research and Development Center, Taiwan, established in 1977. They are maintaining tomato, onion, peppers, and Chinese cabbage. International Center for Agricultural Research in the Dry Areas, Syria, located at Syria, established in 1977. They maintain germplasms of wheat, barley, chickpea, lentils, pasture legumes, and small ruminants. And International Center for Research in Agroforestry, Nairobi, Kenya, established in 1977, integrating the trees in land use system in developing countries. As far as Indian institutes are concerned, as well as international institutes are concerned, I have given the and location as well as here. These are more useful for the examination, competitive examination point of view. Kindly go through all these things. And finally, we can see the evaluation of germplasm, and this is being done to identify gene resources for resistance to biotic, abiotic stresses, earliness, dwarfness, productivity, and quality characters, including morphological and physiological biochemical characteristics. It is also used to classify the germplasm into various groups. And after evaluating, the germplasm has to be catalogued, and then it has to be create a database, and then it should be retrieved. And each germplasm accession is given an accession number as given below. IC means indigenous collection, that is the local collection, Indian collection. EC means exotic collection, that is a foreign collection. IW means indigenous wild collection. The wild collections, wild relatives that are being collected in India are given with IW. And information to be recorded in germplasm maintenance records are the species and variety names, place of origin, adaptation, and various features are descriptors like morphological characters, physiological character, biochemical character, including molecular markers. The catalogs of the germplasm collections are published in the Gene Bank book, and the compilation, storage, and retrieval is now done using the special computer programs. So we have seen all the things, and it has to be distributed and utilized for research purpose. And whatever the germplasm stored in either in India or in abroad, and it is a prophylactic measure, I mean supplied to the researchers who are engaged in the research work of a particular crops. Most of the germplasms are used for crop improvement programs, different breeding techniques, and there must be a quarantine. This is more important. It is nothing but a prophylactic measures to prevent the entry of new diseases, insects, weeds from other countries into our countries. So quarantine officers are located all seaports as well as international airports, and the plant materials that will be given for quarantine purposes, it will be grown in isolation to check the entry of new diseases, pest, weeds, and then they issue phytosanitary certificate. This is more important. Then only after getting the phytosanitary certificate, that will be used for breeding program. Thank you very much for listening my presentation. Kindly watch this video. This is more important. Like, share this video to your friends as well as groups, and comment. That is more important. Another important is please kindly subscribe to the channel. Thank you very much.